So in this tutorial, we'll see how to predict the dynamics of a protein structure that we just obtained from the protein data bank by its temperature factor or the B-value. So the temperature factor or the B-value occupies the identifier 11 in the PDB file format if you open that in the text format, and that just follows the occupancy which lies in the identifier 10 of the PDB file. So these temperature factors or B factors basically designate for the movement of atoms, like the more the temperature factor in terms of their numbers, the more the movement of atoms in that region. And this is usually a result of the electron density maps that you see. The fuzzy regions are actually high in temperature factor. So we'll see if we can predict the dynamics of the protein, well, without running a molecular dynamics simulation in reality, and just do a prediction of how much of a movement each of the atoms in residues might have just based on the temperature factor. So for this, we'll use the PDB file that we have already downloaded, which is the 3FFK, and that's the gelsilin actin complex, along with the water molecules and the calcium ions and the ATP molecule together. We'll just go ahead and load the structure first. That's the PDB with the ID 3FFK. I have it downloaded already. So I go to File, New Molecule, and then browse and locate my structure. So you can see I'm already inside my workspace 1 directory, and I have this structure downloaded. So I'm just going to highlight it and then open it and then load it. So you can find that the default representation, as we have seen earlier in the molecular modeling tutorials, that the default representation will either be a line representation or, let's say, a ball and stick representation, which is a CPK representation in my case, and also the screen. So the background is basically white in my case by default because I've changed some of the settings out here. But most of you will find a black background by default, which you can change by going to graphics and colors and then display and background and choose a color of your liking from here. All right, so the first thing that we'll do is represent the protein and the other entities like the metals, the water, and the ATP separately. Okay, just like we did before, with the aspartyl tRNA synthetase system. So we go to graphics again, we go to representation, and you might be aware that this protein is having 4 chains, which is basically 2 different subunits belonging to a single asymmetric unit. So each of the subunits is actually a heterodimer of gelsilin and the actin protein together. Okay, so if we just type in protein here for the representation, and then we change the drawing method to new cartoon like we did before. So you see uh, all the protein residues are represented in the cartoon format or in the ribbon format with the alpha helices and the beta sheets and the turns or the loop regions represented separately. And then I'm just going to change the material just for the eyes, So just going to pull this up a little bit, and it's good to the eyes. So I usually use this AO chalky format. So it has a better representation, let's say, but it's again your personal choice, whatever you want to go for. And then I just change the coloring method to, let's say, chain. So you can clearly see all the chains together. So colored differently. Basically, the gelsilin in this subunit, on the subunit on the left, is colored in blue, whereas the actin is colored in red, whereas the gelsilin and the actin on the second subunit on the right, which is basically chains D and E, are colored in black and orange, respectively. Okay. So, but we don't need this color representation because we are going to color it separately according to the B value or the temperature factor. So I just wanted to show you that it contains all the different chains. So I'm just going to revert back to the name coloration out here. And then we represent the rest of the entities in this PDB file, which is firstly the calcium ions. So I just type in rest name Ca. So Ca is the residue ID, the residue name for the calcium ions. Then I press enter, and as you know, it's just an atom to be precise, so it can't be represented in the new cartoon format. So we'll have to change the representation to a more desirable format, which is van der Waals of VDW. So if we click that, then we'll see that all the calcium ions are represented in the van der Waals format. So you can zoom in and have a look. So in total, there are 10 calcium ions, so 5 on each subunit. Next, we go for the water molecules. Once again, the water molecules, as I've told you before, in a PDB file, does not really have the hydrogen atoms. attached to the oxygen. So it's difficult to resolve in an X-ray crystallography, even the hydrogen atoms. So they basically have the oxygens in there. So if we type in water to represent the water molecules and then press enter, and we are already in the van der Waals representation. And the coloring is done according to the name. And for oxygen atoms, the color is red out here. That's by default from VMD. Okay. And lastly, the ATP. So we type in press name ATP. So these ATP molecules are actually bound to the actin chain, which is chain B in this subunit and chain E in the other subunit. So as you can see, the representation is van der Waals here and doesn't really look good to the eye. So I'm just going to change it to licorice in order to distinguish them from the other entities. So change it to licorice representation and keep the coloring method as name, and we still find it like you can see it. You can kind of make out the difference out here. Okay. All right. So I'm just going to make this a little bit bigger to visualize properly. Another thing is a shortcut for, suppose your molecule, like you have zoomed into it or you have translated it enough that your molecule is out of focus. So what you can do is on your keyboard, you can just type plus, and it will bring the molecule back again to the center of the screen, to center of the display. Okay, so now what we're going to do is we are going to change the coloring scheme for all of them, all of the representations that we have separately annotated here. So starting from the new cartoon representation of the protein, we'll change the coloring method to beta. So this beta coloring method actually stands for the B values. So it's the VMD has this capability, or for that matter, all the molecular visualization programs, SPIMOL and Chimera even, they have this capability to, consider this identifier 11 from the PDB file and take the numbers or the values that corresponds to the atomic B value. So the atomic temperature factors are taken into consideration, and based on that numbering, the coloring is done. We'll come back to the coloring in a minute, the coloring scheme. First, let me color all the other entities together. So next is the calcium ions, and we change the coloring method again to beta. Then for the water molecules, which are basically the oxygens, coloring method to beta again. And then finally the ATP, coloring method to beta. Okay, so we have everything colored according to their B values. Now the coloring scheme. So the coloring scheme here is basically red, white, and blue, which means red stands for the lower values of the B value or the temperature factor, whereas white stands for somewhere the middle grounds of the temperature factor, whereas blue color stands for the higher values of the B value. So as you can see, the regions that are colored in red are actually less prone to fluctuations. That is, they're dynamically stable, let's say. And the regions that are colored to white are somewhere in the middle, and the regions that are colored in blue are highly fluctuating in nature. And right after looking at it, I mean, right at the first instant, you can make out that the regions that should be fluctuating more in nature, that is, they are not deeply colored in blue, slightly lighter shade of blue, which means that the structure is very, very stable and it's not fluctuating. So when you run a molecular dynamic simulation on the structure, you will find that the structure is very, very stable because first of all, this is a globular protein. And secondly, this has a lot of secondary structures in it, like beta sheet and alpha helix. So that makes it a lot stable than a structure, let's say, which has intrinsically disordered regions in it. Okay. But now, if we have a look at the specific regions which are colored in blue and the other regions which are colored in red, let's say we first look at the protein. So we are going to deselect or deactivate all the others because it's kind of a distraction with the atomic representation of the calciums and the water molecules. So just deselect all of these together and just have the protein in view. And we see that the regions encompassing the surface or the interface between the gelselin and the actin is highly, highly stable. So if you go back to the system, like the description of the system that you have on the slides, it's mentioned there that the gelselin and the actin complex can dissociate from each other by binding of a ligand to the gelselin loop region, which is basically here for this subunit, as you can see, this loop region out here. Just going to bring this more into focus. So this guy. So this is where the ligand lysophosphatidic acid, or LPA, binds to the gelselin um, monomer of a single subunit, whereas the same you will find in here. Okay, so this is the loop region where the lysophosphatidic acid ligand binds to this other subunit. So, and this is the whole molecule of gelselin. So two structured regions connected by a loop region in between. So the interface between these two, the gelselin and the actin, these two interfaces, or for that matter, these two interfaces in the other subunit, don't know whether you can see it properly, but I think you can now. So these two interfaces are highly, highly structured, as it seems from their, the coloring according to the B value, which is red in nature. Especially the actin regions are very red in nature, which means unless the ligand binds to this gelselin binding site, the PIP2 binding site of gelselin, the gelselin and the actin complex will not separate from each other or not dissociate from each other. And since this is not a ligand-bound structure, that is, this is an apo structure, which means the gelselin and the actin complexes will stay together. Okay, so this is like a proof of concept from the mechanistic point of view as well. All right. So apart from that, you find these regions which are exposed to the solvent to be slightly fluctuating. Okay, you have to keep in mind that the beta values, or the temperature factor, usually ranges from 0 to 150. But if you go back to the PDB file in your text format, you'll find that these values are actually ranging up to a maximum of, let's say, 60 at the most, not more than that. So they lie somewhere in the middle. That's why you don't find these the high beta value regions, the highest beta value regions, to be, that bright a blue. So which means they're fluctuating, but not that much. So these regions are susceptible to fluctuation or dynamicity because they are exposed to a solvent, whereas these regions are also lying in the periphery, so belonging to the gelselin. And in the other subunit, you don't actually find that much of a fluctuating region. So the fluctuating regions are mostly belonging to the gelselin subunit, and the actin subunit is actually very, very stable. Okay, so let's turn this off, the new cartoon representation, and let's turn on the calcium ions. So you see there are 10 calcium ions. Once again, these bit 1, 2, 3, 4, 5, 5 of them belong to the left subunit, subunit 1, and the 5 others belong to the right subunit. And you see that the left subunit calcium ions are less fluctuating than that of the right subunit. And then if we turn on the water molecules, for that matter, there's a lot of water molecules, but you can vaguely figure out, particularly this subunit is slightly more fluctuating because of this blue atomic representations out here rather than this subunit. So the water molecules, some of these water molecules are also bound by the coordination state with that of the calcium ions. So each of the calcium ion sites have a single water molecules attached to it. And lastly, the ATP. Honestly, I don't think you can find much of a difference by just looking at it. So these are very, very stable because, first of all, they are bound to the actin protein. And secondly, they're also bound to another calcium ion on that site. All right. So that's that. And the next thing that we'll do is we'll try another method to show the beta values, that is the fluctuating um, regions or the atoms or the residues in the complex. So another way to do it is just go to extension and then you go to analysis and then you load the sequence viewer. So this is from the perspective of the amino acid sequences that are there, or for that matter, the metal ions and the water molecules that lies in the sequence. So if you click on it, it'll open the sequence viewer, and just I'm going to turn back on the protein representation now so that you can compare and correlate with the colored p-values. So you see in the VMD sequence viewer, you'll find all the sequences that are listed. So starting from gelselin, which is at residue position 28, and until, so that's chain A, chain B, chain E, that's for the proteins. And then further down, if you go down, 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 and you have one ATP molecule and all the calcium ions also designated around here. So all of these are represented by the color code of this column out here, which is the B value. So the B value here ranges from, as I said before, from 0 to 150. But you see, most of the colors barely reaches up to the scale of, let's say, 75 even. So after once it crosses 75, it would uh, go down towards the red shades, but it doesn't actually. So most of them lies around the green shades out here. So you see, so within those green shades, there are a few regions which are darker in shade, as darker green in shade. So initially the residue starting from 28, so that's the N terminal of the gelselin, you'll find that it's slightly darker in shade, which means those regions lie in the, the higher beta value regions. So if you just click on one of these, it can represent that particular residue in a licorice format and color them in yellow on your uh, on the viewer. And if you further select this and drag along until, let's say, where this ends out here, you'll find all of those residues that are represented in licorice. So that's great. That's like another validation check to see, just to see whether the annotations by the V values, the coloration by the V values matches up with the coloration that we have with the sequences. And they do. So you find all these regions blue in color, which means these are fluctuating in nature. And that's why they have the shade of darker green on the sequence viewer. Okay. So you can deactivate this by clicking anywhere else. And now we look for any other region which is very, very light in shade, light shade of green, let's say. So select the proper region. So let's say this region out here. I'm guessing this lighter shade of green will be highly structured, and this will probably be buried inside somewhere around here, and therefore they would be less fluctuating and the dynamicity would be restricted in that manner. So let's go ahead and select that. So you just left click and drag along, and it will select the number of residues. Okay, not bad. So this is lying somewhere around this region. After all, this was not a very light shade as we have expected. So guessing by the shades of red out here, imagining still a lighter shade of green would be available to a region corresponding to this out here, which is darker in shade. Okay. So you can correlate and make an assessment of the different regions that might be involved in binding as well. So the more the fluctuating the region, the more prone they are to bind to a particular ligand or any small molecule that might come in its vicinity. And this is also a check before running your molecular dynamic simulations, just to see how strong hold of a structure your protein is, so that once you run the simulation, you can have the atomic fluctuations and compare them with the temperature factors in order to reach to a strong inference or a conclusion. Thank you.